Great, thanks. It's such a pleasure and honor to be here. So thank you so much for inviting me. I'd like to take the next half an hour or so to just talk through some of the work we've been doing on this, what I recognize as a very broad topic. And I've put my contact information here. Should you want to contact me, it's pretty simple. It's just my name with a dot in between at my organization. So no need to even write it down now. You can all remember that. And I'll be in the Slack as well. Let me move my own picture up off of my slides. Okay. Uh-oh. That's a problem. Well, so my slides are showing, but they're blank. So that's strange. Let me just escape out and see what's going on. I've never seen that happen before. Okay. I'm going to stop sharing and see if I can problem solve this on this side. I promised Ito there would be no problems with me giving a live presentation, and now the technology is completely making a liar of me.
So one solution, Lara, may be to save it as a PDF and reshare that.
Yeah.
You're in a conference of troubleshooters here, so everybody's suggesting something on the chat.
Oh, really?
Yeah.
You know, one of the things I found as soon as I became president, I became immediately that day the least capable technologist in the room, and it was like, you know, how everybody sort of laughs at how the person in charge can't actually get anything to work. Like, it just happened overnight, all of a sudden. It was very humbling. Okay, so I've just gone back to an older version of my slides, and I think that this is going to work. So let me try that. Let's see. So. Okay, look, now with pictures. Okay, so rolling back, data intensive biomedicine open practices. I feel very lucky to be in science right now. There's been such a wealth of new technologies over the last decade or so throughout my career that have allowed us to start to capture such intricate, exquisite data on humans, both at the molecular level and also at the level of the person. And so as we move forward with increasingly moving from sequence-based to image-based solutions on the molecular side, and then on the level of the person, moving away from a world where we're simply collecting data from humans when they come to a clinical research clinic to give a single sample at a single time, into being able to collect data from them in real time, outside of the clinic as part of their lived experience, whether that's electronic medical records or sensors on the body or in the home or environment, or increasingly really this, this concept of lived experience. What does it mean for a person themselves to be healthy or diseased? There are gobs and gobs of data coming along, and all of these require us as bioinformaticians to be thinking about how we effectively process that data, analyze that data, and interpret the results. And it's continually true as a new data source comes available, or as we start to integrate in novel ways across data sources, that the methods that we're using are emerging. We're building new methods, and they are not standardized. And they actually are often also not objectively analyzed. And so there's a lot of sort of homebrew methods out there that that people are self-assessing for their efficacy, and it can take quite a while for when each of these new technologies comes forward for the field to sort of standardize around what best practices are, what appropriate methodologies work. And we see this right now in the field, in many places, but in particular, right, the new hotness is the use of machine learning or AI. And we see this in all aspects of what we do. There's great hope, there's great benefit of using methodologies such as this. However, these methodologies, we have the same problem with these as we do with any of the other algorithms that we're building, and that we need to hold ourselves accountable to ensure that we're using them effectively and appropriately, and that the outputs that we have are being, correctly interpreted. And there really has been a bit of a replication crisis here, sort of propagating what we continually see as we move forward in bioinformatics. Where there's a sort of fuzzy line between real breakthrough and what this article terms product showcase. So open science approaches are an obvious solution to this. I think everybody on this call probably knows intuitively what ingredients we need to have robust computational biomedicine. They are founded in the principles of open science. We need access to data, and the data needs to be high quality, and not just generally high quality, but actually explicitly of good quality for the purpose you want to use it. We need to be able to objectively evaluate independently and transparently the performance of algorithms. And then this third point I think is really important as well. We need to be able to independently evaluate not just algorithm performance, but also interpretation of the results. And that can often require very complex, multidisciplinary teams of researchers coming together to do so. So I'd like to direct people to this wonderful article that came out a couple of years ago that looked at the act of doing science, and how we as scientists interact and with what goals. And the long and the short of it is that small teams of researchers tend to innovate. They tend to be seeking new discoveries and on the bleeding edge of knowledge. Well, larger teams come together to actually develop, to take those innovative new ideas and sort of harden them off. And this is absolutely essential in translational medicine, which is really predominantly where we focus our own research efforts right now, where we need to be able to develop really robust outputs, because what we want to do with them is get them into advancements in clinical care. And so you need to innovate, and then you need to harden everything off, and then push it into the translational pipeline. Both of these sets of researchers, whether you're on the innovation side or you're on the development side, they all need access to good quality data. And increasingly for the types of research problems we are looking to solve in computational biomedicine, where we need a lot of data. And so how do we see this playing out? Well, scientists already work in trusted communities. We are right now talking to each other in the context of a trusted community, actually two trusted communities who are coming together. And we see what I'm showing you here is a whole series of consortia. These happen to be ones that SAGE is actively involved in, but it's just a tip of the iceberg in terms of projects where multiple scientists are coming together. And within the context of these projects, people are doing these activities already, these three activities I just talked about. They're sharing data, they're independently evaluating each other's algorithms, and then they're coming together around the interpretation of the results. But they're often doing it in fairly qualitative ways, right? It's through discourse such as these, where we hear each other's ideas and we evaluate them and we make our own opinions about them. And as Ido mentioned, there's increasingly this wonderful community or set of interrelated communities who are moving past this sort of qualitative approach to evaluation to do a much more intentional, independent, objective evaluation of algorithms. And so these are the challenge frameworks, right? Including the critical assessment programs. We are actively involved in the DREAM challenges, as well as we provide infrastructure support to a series of these other groups as well. And what we see consistently across these groups is that their goal, as I said, is to come through and benchmark a field. What is possible right now, given existing data and existing algorithms? What can we, what kind of performance can we get, and where do we need to improve? Whether that is in access to additional data or it actually is in algorithm development itself. And so the underlying premise of each one of these groups, and in fact, other maybe less formalized approaches to the evaluation of algorithms, is accessibility to data. And when we started at Sage, we believed that the most simple and effective way to do this was simply to put all data in the open domain. So that there was the broadest possible accessibility to it, whether it's for these specific types of community evaluations, or whether it's actually for all of those individual groups that are working on innovation to use it. We need the resources to be available to as many possible people for as many possible uses as can occur. The problem with human data is that most of the data we need to answer impactful questions is simply not open data. And there are more than one reasons for this, actually. I'm sure there's lots of reasons for this, and I'll talk about two of them. The first one is simply that human data can't always be openly shared. And this is for reasons of privacy and security. As we continually go into looking at data collected through clinical practice, and in this case, I'm exemplifying this with a project we performed using mammograms. This data has not been consented to be openly shared. It was actually consented in a very specific context around receiving clinical care. And so we need to think of other methods if we want to be doing the sorts of work that I think we all do on big, meaty problems that can actually impact clinical care, then we have to get around this problem of wanting to only work on open data, because in fact, only a tiny amount of the data that we need is and can be open. There's another reason that we are increasingly running into why data is not being openly shared, and I think it's best exemplified by this example. So as the pandemic started and COVID started racing around the world, there was this wonderful opportunity to evaluate the virus as it was moving and as it was mutating. And so this leveraged the open viral sequencing database that already existed. So all of the infrastructure was already in place, and it was, and as well as the common social practice of depositing viral sequencing data into the database. And so the data was immediately available for everyone across the world to analyze, and we could use this data in both our public health and our clinical health and our treatment development programs. And so amazingly important. But what we started to see, and probably some of you ran into this article and this commentary this spring, was in fact, although everybody was equitably putting the data in, the utility or the ability to use the data was not equitable. And so scientists, in this case in the global South, were saying that the open access movement was actually leading to them having less opportunity to analyze data because they didn't have the level of resources that scientists in wealthy nations had to actually use all of this really rich data. So they were contributing data equitably, but not actually getting the opportunity to access it equitably. And so these sorts of things need to be considered as we think about how we share data and how we use data. The good news is that over the past, say decade, there's a whole host of other approaches that have been developed. The ones we're closest to are listed here, but this is by no means a complete list that allow us to access data to perform what we call open science, right? Open analysis on data that is not entirely open itself. And each of these methods differs in their technological approach and their use of data governance policy. I like to think of these as fit for purpose data sharing methods, right? And so you need to match the approach you're going to take to the characteristics of the data, the ability to actually share that data, and the needs of the scientific community that you're trying to enable. And so, you know, on this particular graph, we've shown 2 axes: availability, that is, how many individuals are going to actually be able to access the data, and freedoms, which is to say, and how much freedom do they have in terms of the kinds of analyses they can do. And what I'd like to highlight here across these many different approaches is one example, which is the model to data example, which is in the upper left-hand corner. And so here, as actually with many of these approaches, the entire model is predicated on the concept of a data steward who brokers the relationship between data donors and data users, setting a clear set of policies and understandings with the data donors about how the data will be shared and accessed and used, and then propagating exactly those policies through to the data users. And so in the model to data approach, original data comes from a series of donors. In the case of the mammography challenge, it was mammograms coming from multiple medical centers across actually both 2 continents, North America and Europe. The data is held in a central place by the data steward. Data users are able to access synthetic data and perhaps a small training set, but the actual bulk of the data actually stays in the hands of the data steward, and data users use containers to send the algorithms to the data so they can get some information back, but they never actually touch the data itself. We did not invent this. We are not the only ones who use this, but we find this to be incredibly effective, and in particular when we're trying to run crowdsourced benchmarking programs, such as in DREAM or some of the other critical assessments. So an example, pre-pandemic, we were setting up a system to run challenges, crowdsourced challenges on electronic medical record data. And the basic premise was that we could actually extend the model to data approach to do so in a federated manner where research data was housed at multiple medical centers across the country. And we could access it using the model to data approach to run a challenge where we could look at the accuracy of EHR algorithms, and we could look at how those compared across multiple medical centers. We put this in place, and we started by running a very simple prediction challenge, the patient mortality challenge. One of the things I really like about this challenge is that we use the electronic medical record that exists today as the training data set, and then the traits, in this case mortality, that we're looking to predict is in the future. So we're trying to predict future events within individuals within these medical centers. So how does this actually work? Well, it's a 2-phase submission in this case. The challenge participants will submit their algorithms through the system that we've built called Synapse, which can manage the algorithms and submissions. Their algorithm is first sent to a set of synthetic data that was created by this team and is housed actually in the NIH at NCATS, where their algorithm can train, and they are able to, from that, get back some information specifically about the model and its performance on the synthetic data. So this can be done without exposing any of the data itself to the individuals. Once they're ready, they then actually submit their formal model, which goes into the medical center system. So the example here is the UW medicine system, where it is trained on the actual data, and then the model that is developed there is evaluated on a second set of data, and the users don't actually get any information back about that specifically themselves in the form of an output file. but instead their prediction accuracy is put on an open leaderboard. And so in this way, we're able to objectively and openly evaluate algorithms across the field without actually exposing the data that can't be exposed. We're able to do these sort of open assessments on closed data. So one of the things that was really serendipitous about this program I just told you about was that we'd already set up all of this infrastructure and all of the data use terms when the pandemic hit. And so we were immediately able to pivot the whole system to start to look at additional challenges related actually specifically to COVID-19. And it was a long time ago and a lot has happened in the last year and a half. But when we first started doing this, we are in Seattle. And Seattle was one of the first places that that COVID really started to hit back in the beginning. And so we were able to quickly spin up this program within the University of Washington Medical Center system and expose that data so that the algorithms and the outcomes actually could be learned from across the country as of course the virus moved. So we've used this model to data approach across lots of different challenges, not just with electronic medical record. In each of these cases, there was a reason, in my opinion, a legitimate reason why the data couldn't actually be shared. Some of it is proprietary. This has turned out to be a really nice way to get companies who have really rich data sets to expose that data for use by the research community, et cetera. So I want to pivot here to say, you know, we've spent a lot of time thinking about how we as computational researchers can satisfy ourselves, convince ourselves that our computational algorithms and our results are sound and believable, right? To provide confidence around the outputs that we're developing. But what I think that we still struggle with, and I don't know if you have this this issue, but I certainly see this in every project I'm in, is once we have what we consider a sound computational result, it can often be very difficult to convince scientists in other disciplines, in particular those who don't have computational training and therefore themselves don't have the ability to evaluate the soundness of the results. It can be very difficult to convince them to actually act upon these computational hypotheses that come out. And so how do we start to bridge that gap across disciplines? And what I'd like to do is to give you an example, talk a little bit through a vignette of where how we're starting to think about this problem effectively through a use case. And so the use case is this. Alzheimer's disease, target development. So Alzheimer's disease is a debilitating disease for which there is a paucity of treatment options. In fact, until the famous or perhaps infamous authorization by the FDA for Biogen's aducanumab just a couple of months ago, there wasn't a disease-modifying treatment available. And one could debate that, and lots of people are debating the efficacy of aducanumab. So why is this? Well, essentially it's because the biological mechanisms at play here, they're just not well understood. What we do know is that Alzheimer's disease is a disease defined by pathology and by clinical symptoms that are really pretty general. So in fact, we're probably looking at a heterogeneous set of biological dysfunction that are manifesting in a very simple way. And so when we sort of aggregate data in the typical way, it's pretty muddy because patients actually are highly much more individual than we're giving them credit for. And so what we actually see in the field is that clinical trials continually are looking at the same targets over and over again. And this is because the nature of Alzheimer's disease itself means that the clinical trials to study Alzheimer's disease are exorbitantly expensive because they have to be really long. And so people are making conservative bets. So I don't work in the pharmaceutical industry. Why am I talking about this at all? And that is because the drug industry really builds its target list based on what happens in academic discovery. And so I'm showing you a graph at the bottom here. On the left, you can see publications coming out around all of the proteins. This is specifically looking at all kinases. And on the right, you can see patents coming out around kinases. And simply what you see is that where there are more academic publications, that is where industry starts to look for its next hits. And so it's really important in academia that this is the starting point to think about solving this problem of having limited drug targets in Alzheimer's disease. The great news is that the Alzheimer's disease is a problem that that many people are aware of all the way up through Congress. And so we've seen this huge increase, and this has continued in funding specifically for Alzheimer's disease to the NIH. So how do we actually make sure that this funding turns into new targets? How do we diversify the target pipeline by uncovering understudied biology in this field? And so one of the first things that the NIH did was to develop a program that brought computational approaches, specifically building disease models on human data, into the forefront. And so this is a program that's actually 7 years old that was explicitly designed to generate multi-omic data coming from human brain samples related to disease, and then to evaluate methodology, systems biology, and increasingly machine learning methodologies that can be used to uncover the biology at hand in the disease. The little graphic is meant to indicate that this partnership involved 6 academic groups, all of whom are experts in these sorts of computational approaches, and 4 pharmaceutical groups who were involved as funders, but also as guides to say, hey, you know, you're finding all of this really interesting biology, and I can tell you that when we think about developing drugs, like the first 3 things you're looking at will never be able to drug, but the next 3 things we would be able to drug. So maybe you could focus your energy and time there, which has been amazingly effective. Sage got involved in this project for the second point here, which is we were brought in to work across these multiple computational groups to do an objective evaluation of their computational target predictions using open science. And so the approaches are just as I talked about earlier, right? We made sure that data was being shared early and often, not just with the people in this consortium, but in fact, broadly for anyone to use. In this case, actually, there are no limitations around the data in terms of sharing. And so it is openly shared for anyone, which is great. And then we also came in to start to do this sort of objective evaluation of the algorithms that were being used. In this case, we ran into a problem that we often see when we're trying to do discovery biology, which is that there's no gold standard. So we couldn't run a crowdsourced challenge. We didn't have a gold standard to compare by. And so we had to think about other methodologies we could use to try to do some comparative benchmarking. And so what I'm showing you here is the output of a meta-analysis we performed, which used one of the leading network biology methods from each of the groups involved, so 5 different algorithms, and ran those across all of the data sets that we had access to, which turned out to be 4 data sets, each of which was omics data from human brain samples in the disease and the undiseased state. And we looked to see where the consistencies were in the biology that was being highlighted as highly relevant to disease across this matrix. And so what I'm showing you on the right in this simple heat map, this is looking at the modules and biologies that came out of these analyses. And we see that there are 5 main categories of genes, which are highly related, highly changed with disease. And what I can tell you is the top 3 of these have been well studied in the literature over many years, many decades, but the bottom 2 have not. And so we see these 2 emerging areas of biology consistently seen across data sets and methods, which is a great indication that this is a place that we should be studying. And so each of these groups are very keen on looking in these new areas, and they're starting to partner with experimentalists to try to convince them to start to follow up on some of these hypotheses with using experimental approaches. But here we come back to another sort of systemic problem that we have with the way that we've set up our scientific enterprise. And that is simply that scientists are resistant to studying biology that has not previously been studied. So why is that? You know, we as scientists like to think of ourselves as curious about the unknown, and that is true. We all are. However, we also like to be funded, and we like our research to be published. And the peer review process, particularly at the level of grant review, but also at the level of manuscript review, is biased towards research programs that are iterating around known biology, which is a much more conservative bet, but it's a sure bet. It's in terms of output that will come from research dollars being invested, than scientists who choose to study something that's completely unknown and for which we can't judge whether or not their analysis will lead something fruitful. And that's just simply the way our system is built. And so I can exemplify that here by showing you again data coming from the kinase literature, although we have repeated this analysis looking at targets in Alzheimer's disease, and it looks exactly the same. What you can see is that before the human genome, the only kinases we knew about were the ones we'd happened to purify ourselves. We were able to sequence and purify, and that was very few. And so you see that publications were really focused on those few things which we knew. But the human genome allowed us to then identify all the kinases that existed, that exist in the human genome. And one would think that would lead to a huge uptick in experimental evaluation across the board. But what you can see nearly 20 years later is that's just not the case. We continue to focus our scientific activity on those things which we actually already knew before. And it's just the nature of the system we have. So how do we change this, right? I just told you that we've spent 7 years using these de novo data dependent or data specific approaches to identify interesting new biology related to Alzheimer's disease that may actually lead us to wonderful new drug targets. How do we actually convince experimentalists to do the necessary studies on these understudied targets to move that forward? And to do this, we've actually partnered with a group, an international group called the Structural Genomics Consortium. This is led by Al Edwards at the University of Toronto, although it's across, I think, 5 continents. It's a wonderful international group of structural biologists and medicinal chemists. And what they realized about a decade ago was one of the reasons that experimentalists do not move into new fields is because the activation barrier is really high. If you're moving to work on a new protein, you have to build all your reagents from scratch. You have to build antibodies, you have to build knockout lines, you need lead compounds, you need small molecules that can inhibit, and all of this is very timely and costly to develop. And so their approach was simply to take the open science approach that we've all been thinking about in terms of computational research and flip it into the experimental domain. So they have been generating well-validated reagents for 10 years, including structures, crystal structures, purified protein, plasmids, antibodies, and increasingly also using medicinal chemists to build lead compounds, so small molecule inhibitors, and they are putting them in the open domain for anybody to use, free of any encumbrances, including for the small molecule inhibitors, free of any patents. And the idea here is that they're not trying to build new drugs. What they're trying to do is to build research tools that can catalyze the community to look quickly at at understudied biology and de-risk it for the rest of the community. And it's worked incredibly well. So they've got, thousands of publications, and actually at this point, 10 years in, 50 ongoing trials coming from the output that they've put in. And so we've partnered with them to bring together the Alzheimer's disease target development side of this and this open experimental side. So open bioinformatics paired with open experimental reagent development to try to blow this open. So so this group of individuals is simply taking the structural genomics consortium approach and applying it to the interesting proteins coming out of those computational analyses I just showed you about. So we can encourage the community to take forward our hypotheses. So this is the work, I forgot to put the attribution on the bottom, I apologize. This is the work of Greg Carter and Greg Carey at the Jackson Labs, and then Jesse
Lara?
Wiley and yeah.
Sorry, Cito. I've just been informed that we need to wrap this up. I'm not exactly sure why, but we are already almost into overtime.
Oh, great.
I'm sorry.
I can do that in less than 1 minute.
It's better than getting kicked out brutally, and we do have quite a few questions for you, actually.
Oh, great. Well, then let me just say, and I'm happy to answer the questions in Slack afterwards if we don't get to them all. Just to say, we've built a pipeline to identify the targets. We've picked targets which we are building open resources around and distributing, and we are creating an interface for the experimentalists and the computationalists to exchange ideas, results, and reagents. And I'll end there to say, we hope to use open science to sort of bridge the gap that we see in translational research. And rather than talk through this, I'd much rather answer questions. So thank you.
Okay, thank you very much, Lara. I'm not exactly sure why, but we are basically would have been kicked out in less than a minute from now, and they gave us an extra 5 minutes only. Oh. So there is this question. There's a question by Carol Goebel that generated a lot of interest on Slack. So I think I'll leave that actually for last. And maybe we can go to the Cafe Connect or something. There is a BOSC Cafe Connect, right? Nomi? Hang on. Let me try and get. So let me start with a few questions here. Ian Simpson, for the dream synapse example you gave, what were the challenges generating the synthetic data? If that data isn't sufficiently representative, it would presumably have a major impact on the developed models.
Yeah, I think that's a very good point. And the synthetic data generation, it's an active area. It's an area of active research, and that was part of what we were trying to test with this. Was the synthetic data going to be sufficient? I'm not sure that I could speak to explicitly what the impact was of the accuracy of the synthetic data on the performance or non-performance of the methods, but for sure it's right there in the mix.
Okay, and I will ask the question by Carol. Hopefully this we will be able to stay here a little bit, because this seems to generate a lot of interest. So GISAID is favored by submitters over ENA and other resources because it prioritizes contributor credit, but the GISAID licenses actually don't enable open access, whereas ENA does. Credit was a key message in an earlier talk that was given here that resonated quite strongly. How do we square this equitability circle?
Yeah, that's a great question, Carol. And one that I feel like is, the cafe may be a great place for it, because I don't have the answer to how we square the equitability circle. It's a discussion we need to be having across the field, and I'd be really interested in everybody's opinion on that. Okay. I guess I can say that we've been. I feel like I just didn't answer the question. I don't know what the answer is in this specific case. We've been working hard on how to ensure contributor credit in all of the cases where we're sharing data. And I don't feel like we've solved the problem. We continue to try new things. And I think it's gonna, we just haven't solved the problem yet.